So now I would like to hand over to our Head of Research, Dr. Sarah Schoenberger, who will introduce our speaker for this evening.
Welcome everyone. Thank you for joining us tonight. We're really excited to have Dr. Thomas Park from the University of Auckland join us tonight. And he is part of the Neurosurgical Research Unit, where he's a Senior Research Fellow, and that's part of the Centre for Brain Research at the University of Auckland. Thomas has got a PhD in neuroscience from the University of Auckland, and back in 2015 he got a Neurological Foundation small project grant to start his research into brain tumours, and the whole team and the research has really grown from there, and he's had 2 large project grants since, and he's going to be talking about all of his research tonight. So welcome, Thomas.
Sorry, I'll mute first. There you go. Good evening, everyone. Thank you very much for your time in attending this talk. It's my pleasure to be speaking at the Neurological Foundation event because, as Sarah said, I've been greatly indebted to the Neurological Foundation and their supporters for their continued support for a lot of my projects, 3, as Sarah mentioned, as well as 2 that I've been co-investigators with. So it is really my pleasure to be sharing some of this information and our progress with you tonight. So just going back to my title, it's sort of in 2 parts because I'm covering 2 sort of major projects that had been funded. First is really increasing the effectiveness of radiation therapy on brain tumors. So we really want to be killing the brain tumors as best as possible with radiation therapy. Second is, at the same time, we want to be reducing the radiation-induced effects on the normal brain that is surrounding the brain tumor. So it'll sort of be broken up into these 2 things, and I'll try my best to get this all explained and in sequence. So this is sort of the outline. First is, what are brain tumors? And what type of treatments or therapies are there? But obviously, I'll be focusing on radiation therapy. Now, what is radiation therapy then? And how does this work to treat brain tumors? And then I'd love to share some of our research aimed at improving this radiation therapy. As I mentioned, project 1 is to increase the effectiveness of radiation therapy on brain tumors. And second is to reduce the radiation-induced brain damage and cognitive decline. So, start us off, what are brain tumors? Well, basically, in short, it's tumors found in the brain. Now, if brain cells actually give rise to the brain tumor, we call them primary brain tumors. Here's an example here. This is a glioblastoma, the most aggressive form of a primary brain tumor. And these tumors are known to have risen from the cells of the brain. Now, secondary or metastatic brain tumors here are tumors that have arisen from other parts of the body, like the lung, and they can then metastasize or spread to the brain. And you can see you've got nodules here, you've got different areas where the tumors have entered the brain. So these are known as metastatic brain tumors. Just to go through each type, primary brain tumors, the incidence is about 11 per 100,000. And as you can see here in a schematic, majority are non-malignant, meaning they're low grade. So if they can be treated through surgery and chemo and radiation, and patients have a very high chance of long-term survival. Now, unfortunately, there are still quite a large proportion that are malignant and that are high grade. And as I mentioned before, you've got this glioblastoma, which is the highest grade. And unfortunately, it's also the most common. What are the current treatment regimens for these? I mean, it really depends on the brain tumor type. There are over 100 different brain tumor categories. So they are all slightly different. And clinicians that work with brain tumors or treat brain tumors will have their protocols based on the tumor type. But for glioblastoma, which is one of the main tumors that we'll cover, which is also the most common and aggressive type, as I mentioned, when surgeons can safely surgically remove the tumor, they'll try to remove as much as possible. So here you can see post-operative MRI, that's the tumor before, and this is the tumor, this is the brain after, where the surgeons have taken as much tumor out as possible. But because it's a high grade, you've got tumor cells that are still left over that can't actually be all cut out. So therefore, patients will then undergo chemotherapy with an agent called temozolomide and also get radiotherapy together. Unfortunately, GBMs, or these glioblastomas, always reoccur, meaning that these therapies are not 100% effective. And this means that the overall median survival time is also only 15 months for patients that are diagnosed with glioblastoma. Metastatic tumors, on the other hand, as I mentioned, they have spread from other parts of the body. And to be honest, they actually make up the bulk of brain tumors. Although they don't arise from the brain tumor, there's so many tumors around our body, and 10 to 20% of these adult tumors are known to metastasize to the brain. So that makes quite a large number. You can see here, lung cancers and breast cancers make up the bulk, but melanoma is also there as well. Why those are dominating brain metastases? There are theories, and there's plenty of studies out there, but no clear consensus as to why. Now, again, the type of treatments for metastatic tumors can depend on the tumor type, but as you saw, and as you can see in this picture as well, in the MRI, metastatic tumors are often found in multiple sites because they're coming through the blood vessel and they're landing in different areas of the brain. So this makes it often quite complicated to take out surgically or take all of them out surgically. Big ones might be taken out, but smaller ones, it is not that easy. So chemotherapy agents could be used, but a lot of chemotherapy agents fail to cross the blood-brain barrier, meaning they can't really get to the brain. So this is where metastatic tumors are often treated with radiation therapy, and because they are scattered all across the brain sometimes, they sometimes get this whole brain radiation therapy, which I'll get to soon. So that leads us to radiation therapy, the main topic of this talk and my research with the Neurological Foundation. What is radiation therapy? In short, basically we're shooting precisely, that is, a high energy radiation such as gamma or X-rays, or sometimes high energy particles such as protons at the tumor. And they try to aim it as much at the tumor as possible to slow down the tumor cells' proliferation or growth, or just kill the cancer cells outright. Now, just a little bit of background, but I will try to make it as easy as possible, shall I say. There's two major pathways in how radiation could kill a tumor cell. One's known as a direct pathway, and one's indirect. Direct is where that high energy radiation goes and hits DNA, and that large amounts of energy will end up breaking the DNA. Indirect is where it doesn't hit the DNA directly, but it actually hits water molecules. And as you know, our brain and our body is filled with water. The water then breaks up into a very reactive molecule, which ends up reacting with oxygen again and turns into an even more reactive molecule that ends up damaging the DNA. Either way, both end up damaging DNA to an extent where DNA repair fails. So it becomes so damaged that it fails in its DNA repair. And at that point, a cell will then undergo a cell death pathway or undergo senescence. So they'll stop dividing or they'll die, both of which are beneficial when you're thinking about tumor treatments. Now, these are the types of radiation therapies. First is whole brain radiation therapy. And as the name suggests, it is when, and you can see here in the schematic down at the bottom, it's when largely the whole brain gets radiated. Certain sites might be protected to a certain extent, but the whole brain gets radiated. And as mentioned with the metastatic tumors, it's often used when you've got multiple tumor sites, or the clinicians suspect that there might be undetectable microtumors around the brain. But the bad side is that a lot of normal brains being exposed to radiation, and hence they normally end up with the most radiation damage. Intensity modulator radiation therapy is the one that is most commonly used for primary tumors. And this allows for much more precise control over radiation dose in an area. So you can see here in the middle, it's a little bit more precise in this image. This was the best I could find, which really does minimize the damage to the healthy tissue, but it doesn't absolutely avoid it. So it still has radiation spillover to neighboring brain tissue. And these tend to be conducted over a long period of time. So because they are exposing normal brain, they tend to give low doses over 30 odd sessions, so over a month or weeks over a month. So the patients will have to come in often to the hospital to get this prolonged treatment. The last more common one is stereotactic radiosurgery, where you have a shorter session, so not many sessions. You get really high dose radiation that is precisely targeted. And this has a benefit of having reduced off-target effects, but it's really catered for smaller tumors or tumors that are in precise locations. So if you're not sure where that boundary is, in case of many tumors, this is a bit more difficult to use. So these are your three types that will be most likely be used if you are getting treated for radiation therapy with brain tumors in New Zealand. Now, here are the problems to address, because radiation therapy, although it is really a key and critical tool in addressing brain tumors, it has its problems. One is that high-grade brain tumors, as I mentioned, such as glioblastoma, are resistant to radiation therapy to a certain extent, and they do come back. And as you can see in this study here, even with radiation therapy with drug, most of the patients do succumb to the disease at this stage. So it's not curative. So there's something needs to be done there. Also, as I mentioned, where radiation therapy is effective in reducing the tumor, patients, long-term survivors of these tumors often suffer from brain injuries or cognitive decline. So these are the two that we need to address, and both will be talked about in this presentation. So this leads us to the first Neurological Foundation project grant that we received, and it's really investigating the migratory properties of radiation-resistant GBM tumor cells, right? And as I said, it's aimed at addressing that first problem. Now, just going back and reintroducing GBM slightly, as I mentioned, it's the most common and aggressive malignant primary brain tumor. Global incidence, you can say a rough number around 3 per 100,000. Mean age is in the 60s and affects more males and people of European descent, but it's a global disease. New Zealand incidence and population-wise incidence is not clear. There isn't a central brain tumor database yet in New Zealand, which is surprising, but we are working currently to amend that. So we might have a New Zealand-based database, brain tumor database soon in the next couple of years. And this is the same radiation therapy protocol that I mentioned, so I will skip this. Now, the reason why, and this is the hypothesis and theory why GBM, these high-grade brain tumor cells, seem to be so resistant, are due to the presence of these glioblastoma stem cells. So these stem cells can divide and give rise to the whole tumor mass. So I consider these glioblastoma stem cells as sort of the mothership, and they'll stay out of harm's way. And when they're hit with these radiation and chemotherapies, they're the ones that tend to survive. So the bulk of the tumor will die off, but these guys will survive, and they can actually give rise to a whole new tumor again. And this is probably why patient tumors actually come back. There are theories as to why. These cancer stem cells actually divide really slowly, and if we go back to why cells died in response to radiation, it's because they could not actually repair their DNA in time for their division. So if they're dividing slower, that's giving them more time to actually repair all that damage. And they actually, on top of that, have more DNA repair mechanisms than normal cells. So this is what's making them a little bit more resistant to this therapy. And on top of that, you don't have to read all the articles here. I just have to take my word for it that these cancer stem cells, or these GBM stem cells, not only do they survive, but after being radiated or being selected, they become more migratory. So they actually move faster. And that is quite bad when you're thinking about how radiation therapy is done. So here is just an example of how a radiation therapy could be done by a radiation oncologist. So that's the main tumor area that they find from an MRI or a CT. They will then put a field around it where you get maximum radiation in that area, and then the radiation starts dropping off. Now, imagine if there was a cancer cell there in the boundary, and they get radiation, they survive, and then they start migrating, they move out of that region. That means they're not getting the full dose of radiation. And that could be the reason why a lot of these, after radiation therapy, they find a lot of tumors that have migrated out and given rise to other tumors around the brain. So radiation does work, but this migratory part might need to be addressed. So this is where I was lucky enough to work with a lot of experts in this area. So Emeritus Professor Wilson, Bill Wilson, and Dr. Chorong Hong are radiation specialists, they're cancer biologists. So I knocked on their door to formulate this project. I also work with consultant oncologist, Dr. Han, as well as expert neurosurgeons at Auckland Hospital, Dr. Correa and Dr. Schrader. And I'm also part of the Hugh Green Biobank and the Neurosurgical Research Unit led by Professor Dragunov. But really, most of the credit should be given to the students, the 3 students that worked on this project. So Ramika, Imogen, and Chin. So what do we have? So the CBR Neurosurgical Research Unit, in collaboration with the neurosurgeons, will get donated human brains from neurosurgeries, including GBMs and all these tumors. Over the years, we've been able to come up with protocols that allow us to actually keep most of these brain cells actually alive in our lab so that we can do experiments. And this was published recently in a Nature Protocols paper from our lab, showing that we can basically culture or keep alive almost every cell from a neurosurgical human brain specimen, including here for this topic, tumor cells or tumor stem cells. So how do these tumor stem cells grow once we take it out of a person and grow them in a lab? First, they're these cells that you can barely see, but they start growing in these balls called tumor spheres, like that. And this is basically mimicking how a tumor might grow inside the brain. So they like sticking to each other and growing as a ball. Now, when we label them up, they label up with a lot of the stem cell markers, as I mentioned. So we've got these stem cell balls. And we can even do live cell imaging. So here is a microscope, and we can film as these cells actually move out. So remember, this project really wanted to look at how they migrate. So this is a quick video showing how a patient tumor in our dish is moving out and migrating out. So imagine this sort of being in the brain as they're moving away from their source. So if that was being radiated, how would that affect their migration, moving out? So the protocol or the method to test this was we first needed to select the radiation resistant cells. So we took the same patient case, and for control, we just left them as they were, as you can see there. And for radiation selected cells, we would radiate them 3 times with 3 gray or quite a high or mid-high gray level dose radiation 3 consecutive times to basically select for the cells that will survive this radiation. This is a dose that is much lower than what a patient will be given in the clinic, but this is a cell in a dish which is getting direct radiation instead of going through the skull. So this was enough to actually select for the cells that do survive. So these cells here, you can see there's really quite a difference in how they grow, and they also divide much slower. If you remember the cancer stem cells, these cells that are resistant tend to divide slower, and they really do. Using those, we did the live cell imaging. So here you can see the control and the radiation selected tumors. And after 24 hours, you can already see that the radiation selected cells are moving further away from their origin, right? And this is really profound and clear by the 48-hour stage. But although it's clear, we just wanted to make sure that we weren't biased. So I approached some very talented engineers to see if we can just use machine learning and image algorithms to pick out the migration patterns. And this was done by the PhD student Chun. And we use this to quantify exactly those experiments. And again, as you can see by eye, but as you can also see by a computer algorithm quantified metrics, that when you have the radiation selected cells were migrating much faster away, covering more area than the controls. So, you know, we've got this, and it goes to show that our model is in line with other publications showing that maybe radiation is increasing the migratory properties of these cells. But what might be causing this? Right? We've got an effect, but we need to know why. Now, going a step back, maybe about 5 years back, before I started the neuro-oncology or brain tumor side, I was a neuroscientist that was interested in looking at neurogenesis and how new brain cells are born in the brain and how they move and become new neurons. So I've got a quick schematic. So here in our normal brain, all our brains, even in adults, we've got stem cells, and these stem cells can divide and they replace lost neurons in our nose, our olfactory bulb. And in order to do so, these cells need to move. Now, when they move, they express this molecule called PSA-NCAM. This is not the only molecule they make, but this molecule makes the cells slippery, and that allows these cells to move through the brain. Now, as I mentioned, these tumor cells are moving through the brain. So we were wondering if the tumor cells were hijacking the system that's inside our brain to do the same thing. So we then searched for PSA-NCAM in these tumors. And lo and behold, these tumors that were migrating, they were expressing high levels of this migratory molecule, PSA-NCAM. And we then looked at the radiation-induced migratory cells, and you can see that although the normal migratory cells did express PSA-NCAM, and these are the levels here, when you increase radiation dose, so when you give them radiation, they actually increase the expression of PSA-NCAM. So you can see that quantified here. It's pretty clear by how bright the green is in this picture, and also the brighter band that you see there in a Western blot. So showing that when these cancer stem cells or GBM stem cells get irradiated and they survive, they increase PSA-NCAM, and this might be contributing to the increased migratory movement. Since that could be our target, we found drugs and enzymes that could actually get rid of that PSA-NCAM. So you can see that the green is gone, so we can get rid of that. And when we did so, it actually reduced the migration of these cells. So you can see here, that's control. And when we treated and got rid of the PSA-NCAM, they basically reduced their migratory properties. And you can see this actually shown in cell tracking and quantified likewise. So with that, we've got to a stage where, and this is the part that I didn't cover, I might just quickly cover this because this is all related as well. You can see here in an actual patient's tumor, if you have high levels of PSA-NCAM in that patient's tumor, we found that that patient's tumor was actually larger. So they possibly grew faster. And also those patients that had high PSA-NCAM actually, unfortunately, passed away faster than patients that had lower PSA-NCAM as well. So that all sort of matches up with what we see in patients as what we saw in cells and with our drug studies. So at this stage, the last part of this project was to look at the RNA and the transcriptome. So what the cells, after radiation, how are the cells changed? Not just PSA-NCAM, but all the other genes that might be involved in migration. How are they changed? So we've extracted that RNA, and we've already basically paid for that analysis, and that's currently in the works. It would have been nice if I had that to show you, but maybe next time I could complete this story. But that is currently underway. And this is an exciting, another project that is about to occur. So this is in collaboration with Dr. Ramesh Pandey. He's a radiation oncologist, the actual people that do radiation therapy. Professor Wang, who is actually an image analysis engineer and AI expert. We're also trying to see in actual patient brain whether we can predict where the cells might spread in response to radiation therapy. That means we can basically block their path. Its tracks if we actually find this. So it's a really exciting development project. And again, if I'm invited again and at a later time, I'll be hope we can share good data with everyone. So that leads us to the second part, the second problem, which is where radiation therapy was effective in reducing tumor, that's great for the patient, but those patients then ended up suffering long-term brain injuries and cognitive decline. So we want to, again, mitigate that. And again, no need to really read all these papers. They're just showing that that is the case, or they're reporting that dementia and cognitive decline happens after radiation therapy. So what are some possible solutions, even before we get into the research? So this is a relatively new development called proton beam therapy. So this is radiation therapy, but if you look here on the side, conventional radiation therapy shoots beams of radiation that will go through the tumor, and the radiation oncologist will try to aim it so that multiple beams all converge onto the tumor, creating the highest dose on the tumor, but you still have these other areas that are still getting exposed to the radiation. Well, here, proton beam, they shoot protons and they basically make it stop inside the tumor. So it basically stops there and releases its energy. So it basically reduces the radiated normal tissue, so amount of normal tissue being radiated. Now, it sounds really good, but it's an extremely expensive setup. Only a few places around the world has it. I think none in Australasia at the moment, or there is one being set up in Australia, but one unit. So it is extremely expensive. Multi-million-dollar facility is required. So currently not available in New Zealand. Now, there are radiosensitizers, and this is used for peripheral tumors. So these radiosensitizers are compounds that basically act on pathways that allow cells to become radiation resistant. So they block those things, and that makes the cells more vulnerable to radiation. And the problem is it's working fairly well in certain tumors, but the problem is these compounds, not many of them are brain penetrant, meaning they can't really get into the brain very well. So therefore, we basically at this stage, in many centers around the world, you really need a way of protecting the normal brain. So we need possible brain cell protecting agents so that when radiation is given to the tumor, the surrounding brain might be protected. So more research, and there are some compounds that are out there that are being used, but more research is required to understand the damaging effects of radiation on these various brain cells so that we can actually find a correct target for protecting these cells. And that's going to be our project here. So just going a step back, many people have looked at acute and gross anatomical changes like radiation necrosis and why that might be occurring. But our work is much more long-term, not big, massive changes where you see edema and cells dying, but subtle changes that occur over time. So that's our second neurological foundation project that was funded this year, and it's planned to start in December, but basically really in 2024. So what I can actually show you is sort of the plan and what we have so far as pilots, and then we'll just have to, I think Sarah will just have to invite me back in a couple of years. So again, I'm really lucky, privileged to be working with great scientists, clinicians, and group leaders, and we are recruiting talented graduate students onto this project as we speak to initiate this project. And as I mentioned before, now we're focusing on all these other brain cells that are not tumor cells. So remember, we could actually isolate and keep alive all the different brain cell types. And because we have this precious resource, we can actually then look at how radiation might be affecting actual human brain cells in a dish. So just a schematic showing that we can actually keep alive most of the brain cells from a human brain when we get the human specimens. And what's really exciting is, for the first time, at least when we published this was the only publication in the world showing that we can actually keep adult human brain cells, neurons, alive and firing. So this is a 52-year-old neuron, much older than me. And we kept it alive outside of a person's brain, and we could not only keep them alive like this, but they could actually talk to each other. So here, this is a patch clamp. And Dr. Kevin Lee, who is a collaborator on this project and an expert electrophysiologist, patch this cell, and you can poke the cell and listen into them. And you can see here, this is what we got. So basically, this is them talking to each other and communicating in our dish. And here, this is just another evidence showing that. Sorry, I'll just turn that off now. So on these cells, we can actually then give radiation. And you can see here at the top, these are our neurons in green, and blue is the nucleus where the DNA is. Now, when you give radiation, can you see those red spots inside the neurons? That is DNA damage. So this is DNA being broken, and gamma H2AX basically picks up broken DNA. So it's good to see that our model, our system, is creating DNA damage inside these neurons, and not just in these neurons, but all these other cell types that we've cultured. We can do the same thing to see how a radiation, DNA damaging radiation, will change these cells, and how that could potentially lead to cognitive decline later on. Now, this is probably the most exciting part of this project. When neurosurgeons have to go in and take out very deep tumors, like here in the schematic, they have to go through some normal cortex, some normal brain, to actually gain access to that middle tumor. And there are some surgeries where they have to cut an entire sort of a big part of the brain's lobe, called the temporal lobe, to cure epilepsy. So when we get those specimens, we actually get normal cortex, or normal brain, not just the tumor. So here's Kevin again, and he's perfected this technique where you can get that normal cortex. So this is a normal human brain cortex, and we can slice it in very thin slices, but this allows us to keep the entire brain architecture. So it's not individual cells like I showed you before. We're actually keeping the brain intact. And we can culture them in these dishes, and we can keep them alive for weeks. And using this model, and as you can see here, they also fire and action potentials, and they can talk to each other, so they're still alive. You can see all the spine's beautiful neuron here with their connections with other neurons, and this is how they communicate with each other. We can give them a neurotransmitter-like signaling. So this is how our brains actually connect with each other or talk to each other, is through neurochemicals. And we can do that. We can mimic that here in the dish. And this is a way of also creating memory. So this is how memories are formed, or skills are learned in our brain. And we can create something similar in a dish. And these are the things that we can use to see how radiation affects them afterwards, right? If memory was formed quite easily prior, but after radiation, it doesn't, that could show that there's something going wrong in that process after radiation. And that's something that we can target. So again, we're setting up a protocol to do exactly this. So we're now giving 3 doses of radiation again, but at a much lower dose, at a dose that sort of equates to about 20 gray. So 20 gray is roughly in this region here, in that sort of green to blue region. That is the region that the normal brain will normally be exposed to when a tumor in the core gets the full blast of radiation. So we want to mimic roughly the radiation dose there, and we did. And this is one pilot study, and I'll just guide you through it. Sorry about the quality of the image, but the red are your neurons. These are your brain cells, and white is your astrocytes, or your support cells. And they're all there. They're nice and healthy in that. And then after 1 hour of radiation, so here we've given that radiation, and it's 1 hour afterwards. Can you see that green? And I've just taken that and split it out at the bottom just to see it better, so at a higher magnification. So you can see that green dot, the DNA breaking, happening inside neurons, happening inside astrocytes, these support cells. And these are the IBA1 microglia. These are the immune cells of our brain, and our immune cells are also getting DNA damage. So all the cells in our brain are getting damaged by radiation. And this is the real powerful part of this system. And this has never been done before, as far as I can tell, in the world, where you get the entire, not just single cells, but the entire system together. Because brain does not act in a single way. They act as a collective. It's a network. So you have to sort of look at the entire network to see the real effect. And what was really exciting about this, and unfortunate basically for the patients maybe, but what we found was after 24 hours, can you hear in this up here, you can see a lot of the green is gone, meaning the neurons and these other cells have repaired a lot of that DNA. damage, potentially. But what has happened afterwards is you can see a lot more white and a lot more fibrous white cells. These are your astrocytes, and this might be an indication that they are slightly angry and they are activated. And this could potentially be neuroinflammation at play. We'll have to see. These are all very early studies, but we're keen to pursue this in the coming years. So with this, the objective is once we go through this protocol, we're going to look at RNA sequencing. So looking at the gene expression of every single cell. So single nuclear RNA sequencing, we'll be able to see how every cell in that brain is responding to radiation in terms of how their genes are changed. We can then validate those gene changes on protein level and cellular level to see how that actually manifested as a function. And as I mentioned, we can also look at how that ultimately changed the physiology. So how it changed how the brain cells function, how it might affect memory formation or anything else that could be recorded. So this is the sort of the project in a nutshell and the plan. And if we find a biological target with all these studies, then we're through another project that the Neurological Foundation has co-funded with a collaborator, we've actually developed brain-penetrating drug delivery systems. Remember, one of the problems was a lot of the drugs could not get into the brain. We've found ways of delivering drugs into the brain, into brain tumors. We can potentially deliver some protective compounds to these brain cells if we find a viable target. And this project here that is also Neurological Foundation funded. This is the magic chemical that can actually transport molecules into our brain, and we can start adding different drugs onto it to make this happen. So in conclusion, our studies really want to increase the effectiveness of radiation therapy by targeting tumor cell migration evasion. Also, at the same time, we want to decrease the off-target neurological effects of radiation therapy by first determining how radiation affects brain cell function in live human brain cells and in whole live human brain slices. Now, with this knowledge, we'll like to develop algorithms and molecules that could maximize damage to tumors while preserving normal brain function to ensure long-term quality of life for patients. On that note, this is sort of our overall 10-year neuro-oncology vision in our lab. So we've got patient samples and we're collaborating with the hospital. Using those patient samples, we're doing all these different screens. With that information, we're working with medicinal chemists and animal in vivo testing facilities to see how they might work. And then that is all learned through computer and AI to then hopefully facilitate some precision medicine and therapies for the patients later on. So again, thank you for supporting our 10-year vision. Neurological Foundation has been critical in supporting the first 5 years of this 10 years. And we look forward to showing you how the next 5 years will pan out. So with that, I'd like to acknowledge first and foremost the patients and families who donated their tissue for research. Without them, none of this is possible. The animals that were sacrificed in this study. Also, my mentors and supporters, Professor Mike Dragonoff, Distinguished Professor Sir Richard Ford, Professor Morris Curtis. Also, previous and current graduate students that really did the heavy lifting of a lot of these projects. So I'm really indebted for all their hard work. And of course, as the saying at the start said with Neurological Foundation, it is not an individual effort, it's a collective effort. It's a collective effort between all the funders and the supporters, as well as all the collaborators ranging from clinicians to scientists to engineers. So really, I'm privileged to work with everyone and be funded and be supported by these institutions. So thank you very much again for your attention and for your generous support. Thank you.
Well, that's absolutely fascinating, Thomas, and so much information there. And I can see that you do a lot of very complex research, and I really appreciate you putting it into something that is understandable for the rest of us. And we're just going to open it up for questions now. So if anyone who's listening would like to put a question in the Q&A, we'll see how many questions we can get through tonight. So first of all, we're going to start off with a question about the spinal cord. As we know, the spinal cord is part of the central nervous system, which is the brain and spinal cord are part of the central nervous system. So where there is a diffuse tumour inside the spinal cord, how would radiation affect that? And do you know whether radiation is used for these diffuse tumours in the spinal cord, or whether it's not an option at all?
For that question, I've really only focused on the brain and not really in the spinal cord tumors. So it won't be my position to be able to tell you whether a certain therapy is possible or not. But as you saw, I collaborate quite widely with radiation oncologists and people that actually do radiation therapy for spinal tumors as well. So if you would like more information, I'm happy to provide those after our talk, but I'm probably not in the best position to comment on that right now.
But if someone was asking their neurosurgeon about that, they would be able to advise them?
Yes, and if they are able to send the question to me, I can easily pass that on to our collaborating clinicians that could easily be able to answer those questions in the most accurate way.
Oh, fantastic. Thank you. So I've got a comment and a question that says, fantastic to see slice cultures up and running, which is where you had whole pieces of the brain that you're working on. How many millimeters are they? How big are they?
All right. Because you've now sliced them, there's no blood supply that is flowing through the brain. So in order to keep them alive, you need them to be fairly thin to allow diffusion to go through. So the nutrients to go in and the gases to also be exchanged. So they're around 200 microns, so 0.2 millimeters in thickness. It can go a little thicker, but the thicker you get, you're now risking the middle part not getting enough perfusion and dying off. So that's sort of roughly where we have it. I mean, a lot of the protocols that I mentioned show exactly what slice thickness we've used to culture these slices.
So it'd be difficult to see those without using a microscope then. They sound pretty small.
I mean, the width is there. You saw it. You can just take it. It's the thickness. It's the thickness that's 200 microns. So they're really thin, but in terms of their area, they're quite large. Sort of your fingernail. So
yeah,
that's your fingernail. It's just the thickness is 200. So that covers pretty much the entire cortex. So you get all the cortical areas. So our brain is divided into different sort of layers of neurons and cells, and you can see all that when you have a cortex size around that big.
So how accessible is radiation treatment for those impacted by tumours? Does it require people to travel to the main centres? Are you aware of how many centres in New Zealand do radiation therapy?
I don't know the entire numbers of centres, but all the major centres, Waikato and Wellington, Auckland, all the Christchurch, the major centres and major hospitals do all have radiation therapy units. I talk with the radiation oncologists in each of those centres on a regular basis. So yes, so you might not find it in very small centres, but any major centres scattered around New Zealand, you should be able to get radiation therapy access.
And how does the brain tissue that you're using fit in with the Neurological Foundation Human Brain Bank? Is it a separate biobank that you're using? And is your tissue accessible for others to research on as well?
So the Neurological Foundation Brain Bank largely is donated brains, so bequested brains. After a person has passed away, they donate their whole brain. In that sense, the human cortex, especially the neurons, normally they die within 5 to 10 minutes of actually losing blood supply. So from a donated brain from a person, it is impossible to basically have that brain and then be cultured within 5 to 10 minutes of that person passing away. So the human brain bank is really not used or can't be used to keep brain cells alive, as in the neurons. Some brain cells can stay alive at extended hours, but neurons are not. That's why we have to work with the surgeons, and as the surgeons are cutting those brain regions out during surgery, we need to basically be there and then get that into a special media, and we need to then actually culture that straight away, or else the neurons and some of the cells will die. Now, for tissue to be shared, it will really need to be spoken with the director of the Hugh Green Biobank and the Neurosurgical Research Unit. So that's Professor Dragunov and Dr. Jason Correa. So they're the directors. So if you are interested, you might need to talk to them about whether it is possible. But at this stage, the cortical specimens are very precious. It's not often that a normal brain would need to be taken out. I mean, the surgeons will try their best not to. It's only when they have to that they get taken out. So it's not a resource that is plentiful, shall we say. So key research questions need to be asked on the brain slices to maximize the output, shall we say.
So you often don't know if you're going to get a piece of healthy brain tissue until the surgery's underway. Would that be correct?
The surgeons have a fairly good idea because they plan their surgeries. So they'll know where the tumor is, they get the scan, and they plan their surgery. So they have a fairly good idea of whether one surgery might have cortical tissue available. As I mentioned, some surgeries such as temporal lobe epilepsy surgeries, so temporal lobectomies, I mean, that's a guarantee because that whole lobe is being taken out to treat epilepsy. So that, yes, but for those surgeries are rare, and it's reduced, and it's on the decline because better drugs, they're getting treated better, which is a good thing. But the tissue that we get from tumor surgeries, it really depends on what type of surgery it is.
So your team has to be prepared, do they, to go and pick up the tumor tissue during surgery when it's
yeah. So
as soon as it's taken out?
Exactly. Yeah. When we first started this, I used to be in scrubs with the surgeons and basically setting this up. Now the system has become a lot more systemized, so it's systemic, and we've got the Hugh Green Biobank has a manager now. That person gets the call, and there's a research nurse on the other side. So it's a lot more coordinated. But when we were first starting this, it was us on the ground doing all this. So yes, we can now do this in a much more systematic way.
And if you use that temporal lobe that's removed in some cases of epilepsy as a control, how much do you consider that to be normal brain tissue? Because obviously the brain's affected by epilepsy.
Of course, you can never say that is normal tissue. There's a reason why that was removed. So normally in most cases, and this is me sort of semi-speculating at the same time as a little bit of an educated guess, is that the epilepsy for many of these cases do start in the hippocampus area. So it's sort of a tissue that is a little bit away from the main temporal lobe area that sort of gets cut out with it because the spread, the seizure spread sort of happens through that pathway. So it might not be the core diseased area, but it's still affected and it's still part of the disease. So you can't really say that's absolutely normal, but it's not a tumor. It's still a brain. But yes, you're right. It is not normal. But as I said, it's not. Why would you take out normal brain? So you've got every model. So we're using it as a control, but we're seeing how radiation might affect that case. So everything should be normalized to the control. But yes, you're right. That control, we always have to take with a grain of salt that it is not perfectly neurologically normal.
And can you talk us through how radiation might affect people differently? So do some people fully recover from the brain damage associated with radiation therapy, or is there different degrees of impairment? So
it's not I mean, the report when they did studies is not 100%, but with brain tumors, as I mentioned, there's so many different types of brain tumors, and also there's brain tumors in different regions. So this is what the radiation oncologists will have to do when they see the tumor, is they draw boundaries. But if that tumor is close to a certain area that is very important in cognitive function, like the hippocampus, they have to try their best to avoid it, and so forth. So there's lots of protocols that I'm not an expert in, but the radiation oncologists are, to mold around key brain regions so that that radiation is not going to cause more harm. But in saying so, because they're in so many different regions, it's hard to tell whether it's that region, it's that person, whether it's that radiation, or a combination of all of them that might be causing it. So we only really end up seeing, did that person end up getting developing cognitive decline after a while, or not? And that's sort of what we end up seeing over time, and we see the results. So this is why I'm really interested in this research, where I'm looking at the small changes, the genetic or like the gene expression changes that occur after radiation that could manifest into potentially cognitive decline afterwards. Because from a lot of the literature out there, neuroscientists know quite a lot, and don't know quite a lot, about how cognitive decline occurs in other diseases such as Alzheimer's and so forth. If per se, say some of these radiation therapies increase or activate some of those pathways that are also shown in other neurological diseases, that could actually give us clues to how radiation might be causing this long-term deficit. But these are all questions. We don't have answers. I'm really keen to look into it and work with the colleagues to answer this.
So is it mainly cognitive decline, or can the radiation therapy affect other parts of the brain like speech or movement?
Absolutely. Yeah, it's just not just cognitive. I just use that term as a loose term. It's brain function. And again, it depends on where the tumor was, and also where the radiation therapy was. So it does affect movement, sometimes mood, and like basically brain function. You could just group it as brain function, and different patients might manifest with different symptoms. But sorry, I just wanted to say that in saying this, I'm not saying that radiation therapy is bad. It's still a very important therapy. It is very effective in treating tumors. It's just that we need to improve it with any technology and technique and treatment. There are side effects. We do want to. We're doing this to improve that. I just wanted to make that clear, because sometimes when I say this, it feels like I'm saying that radiation therapy is really bad, but it's one of the key tools for treating brain tumors and tumors in general.
Yeah, and we always advocate for people getting that advice from their neurosurgeon anyway. So yeah, thanks for just reinforcing that again. The surgeons will always advise people on what is going to get them the best outcomes, which is really exciting. And we saw that recently when I heard a talk from some of the neurosurgeons that you're working with. Their passion for what they do is really amazing. Someone is asking about using a cyber knife. Do you know what that is?
Yeah, it's a much more it's not the it's a much more targeted and radiotherapy that is it's much more similar to a stereotactic, and it's more geared towards a very defined lesion, from what I know. There are many different types of radiation therapy and different iterations or technologies that are used. There's things called pencil beam therapy and so forth. So these are constantly evolving. But what I'm focusing on, and what I've mentioned and showed today, is the general ones, because that's what most people are likely to receive over these more very specific types of radiation therapies or treatments.
Are any of them available in New Zealand? Because I know you mentioned that proton one isn't.
Yeah, I believe it is, because I've spoken to radiation oncologists, and they've mentioned. So yes, I believe it is, but I'm guessing again, this is one of those where you really want to check with the radiation oncologist or the neurosurgeons to see if those are options in New Zealand.
I also noticed in one of your pictures of the neuroinflammation that's going on. Are they testing neuroinflammatory medicines?
Yes, I mean one of the because of the free radical being produced will create will activate inflammatory pathways. So it's a fairly well-known effect of radiation. I'm more interested in how that then influences the long-term effects, but the more acute effects of radiation, where you clearly see damages and edema and swelling and so forth, are largely due to inflammation and immune cells infiltrating in. So they are using certain compounds, and there's lists that people have used to see if they can reduce the free radical production in the brain. Some people have even used antioxidants, vitamin C. So there's things out there that people are trying. As I said, there's no clear consensus as to what is best. And yeah, happy to share that information. There's good reviews out there, so I'm happy to share it maybe with you if there's quite a few people interested, that you can share with people who are interested from your end.
And if people are interested in other remedies, such as homeopathic remedies or other things, how would they go about getting research projects done on those?
I mean, you can. I guess at this stage, if we actually find there's a lot of information on how these homeopathic therapies also influence, but they do influence the biochemistry of cells. That's why a lot of them are used. So if we actually find pathways through our research, and maybe others from around the world, if we find pathways that are definitely affected that could be targeted by those could be an option. I mean, it's rational, but as it stands, testing every one of those will not be an easy thing to do. It will probably need to be more targeted after you find a target. That's my opinion.
And you mentioned the effects of treating the PSA NCAM. Is that something that's going to be looked at as a treatment, or because it also does an important biological role, can you not remove it?
It's definitely a avenue, because if tumors are expressing so much of that, and that's the main problem, that the tumor cells are spreading, then we could potentially target that for the meantime. Of course, that might have slight side effects on stem cells moving, but so does radiation. Radiation also stops a lot of the stem cells from proliferating through that point. But if it's a therapy that you're going to keep for a short amount of time to basically kill off the tumor, and then when they're done, it could be restored, then it could be an option. But again, those are things that you want to look into as the therapy progresses. But it's an exciting area, so I'm looking into it.
Yeah, it sounds like there's lots of exciting avenues to go down, and I guess it's really difficult to choose which one might be the most productive. But we're just going to wrap up with one last question about whether your research includes acoustic neuromas.
I wouldn't know what that actually is, so my apologies. I don't want to be saying anything that I'm not very aware of. Yeah, whoever asked that question, my apologies, but happy to again, if there is anyone around me that knows that disease, or at least that tumor type, then I'm happy to guide you through or refer that person to you.
Oh, thank you so much, Thomas. We're going to have to call it a night now because it's 8 o'clock, but I feel like we could go on talking about this forever, and you're just a treasure trove of exciting information and exciting possibilities. For people with brain tumours, we all know people that have suffered from brain tumours, and we're really all got our fingers crossed for some more effective therapies and getting those drugs into the brain as well. So we're really excited about watching the outcomes of your research over the next few years.
Yeah, thank you very much for your support.
And thank you everyone else for joining us tonight. It's been a really great session, and it will probably be on our website tomorrow. Good night.
Thank you.